Okay. So I'm presenting Derek Applewhite. He's currently an assistant professor at Reed College. He got his bachelor's degree at the University of Michigan in Ann Arbor. He got his PhD at Northwestern University, and he did his postdoc at the University of North Carolina. His main research interest is to understand the regulation of cytoskeleton and cell migration using Drosophila. We're so thrilled to have Dr. Applewhite here for our Diversity in the Molecular Biosciences retreat. He is an active advocate for BIPOC scientists and is strongly committed to fighting against inequalities in academia. He's also the co-chair of the LGBTQ+ task force for the American Society for Cell Biology. And all his efforts in lifting BIPOC and LGBTQ+ students in STEM is just truly inspiring. So anyways, without further ado, please, Dr. Derek Applewhite.
Thank you so much. That was really kind. And thank you for the invitation to speak here. I'm really excited to share my students' work. So I am at Reed College. And if you're not familiar with Reed College, it is a small, let me just stop for once. I have to minimize this. Okay, there we go. It's a small liberal arts school in Portland, Oregon. And this is a picture of our Elliott Circle. It's kind of the main sort of cross path sort of area in the campus if you're driving in, and beautiful sun kind of hitting the light on the cherry blossoms. So this is where I work. And Reed is a small liberal arts school, as I said before. And so I want to share the work of the people who actually did this work, which is my undergrads. I work only undergrads institution, and so Aiden Terrain, who actually is he graduated last year, but he stayed on and is working as a post-bac in my lab. JJ, another student, he's actually a student at WSU, but also does part-time work in my lab. Amy just graduated recently and is now at the University of Washington in a neuroscience program. And Sushruta is a senior this year who's working for me. He's actually a physics senior. So these are people who actually do the work. I'm just their mouthpiece. And please feel free to interrupt me at any time. I'm used to being interrupted. I teach a lot, so I'm constantly fielding questions on the fly. So don't hesitate to jump in at any time. So I'm going to take it really basic because that's kind of where I come from. And so the basic understand. Oh, I didn't even tell you the title of my talk. So the title of my talk is the role of split discs in the regulation of non-muscle myosin 2 contractility. And so maybe many of you might not be fully familiar with molecular motor proteins, non-muscle myosin 2, so I will take a second to break that down a little bit for you. At its basic level, molecular motor proteins turn chemical energy into mechanical work. And there are three main families of molecular motor proteins: dynein, and here we see cilia here, and flagella. Both of these are microtubule-based structures that are moved by dynein, and kinesin, which is, this is a S2 cell that's been treated with cytokeratin D. It's causing these processes to form. And what we're watching is the movement of peroxisomes up and down these microtubule-based processes. So both of these are microtubule-based molecular motors. Dynein is a -N walking motor. Kinesin is a +N walking motor, most of them. There's some -N ones. And then there's myosin. Myosin is an actin-based motor. This is a cardiomyocyte that is contracting on a compliant matrix. And this happens spontaneously in culture with cardiomyocytes. And if this is what we're watching is the activity of a type of myosin. This is more of a cardiac myosin. But nonetheless, myosins are very similar in that they bind to actin, in this case causing contractility. So non-muscle myosin 2, as its name suggests, is found in mostly non-muscle cells, although muscle cells also have myosin, non-muscle myosin 2. And really the main point of non-muscle myosin 2 is to cause cellular contractility. So here we're looking at two cells undergoing cytokinesis. This is classically what we think of when we think about non-muscle myosin 2, is making sure these two cells divide equally. But non-muscle myosin 2 is also really critical to morphogenesis. So this is ventrofloral formation during Drosophila morphogenesis. And what we're watching is apical constriction in action. Apical constriction is just the constriction of the apice of these epithelial cells, going from a cuboidal shape to a pyramidal shape, and that's enough to drive the invagination of tissue, allowing for this morphogenesis to happen. Non-muscle myosin 2 is also really critical for cell migration. So we're watching the spontaneous sort of looks like explosions to me of assembly of non-muscle myosin 2 filaments in the middle of this cell, and this cell theoretically is migrating on a substrate, and these spontaneous sort of contractions are happening as the cell is migrating. And at its most simplest, non-muscle myosin 2 is regulated by a molecular change. So phosphorylation of the regulatory light chain, which is the light blue bubbles here, leads to opening up the molecule. And once it's open in this conformation, it's able to form oligomers, so it doesn't work well by itself. It actually works much better in teams. It's a terrible motor by itself, but when it oligomerizes with other motors, it works better as a team. And so once it oligomerizes into larger structures, these then can bind to actin and then cause the sliding of filaments and leading to the contractility that I just showed you in those movies. And what does this look like? So this is an S2 cell. It's plated on con A, and so S2 cells are draw off the tissue culture cell. They're really, really flat when they're plated on con A, and so this is imaged by TURF microscopy, and TURF is excellent to image these cells because they're so flat. Expressing here is spaghetti squash, which is that regulatory light chain. So that's kind of a proxy for non-muscle myosin 2, and actin. Now, I can't, due to the resolution of this microscope and the microscopy here, I can't for certain say that these barbell-shaped molecules are individual non-muscle myosin 2 filaments that are forming these larger white oligomers in the center of the cell. But if you just zoom in, you can at least think about that intermolecular change that's happening, where it's going from an auto-inhibited conformation to an open conformation, and then into these giant oligomers. So at its base, this is how non-muscle myosin 2 works. Again, it works better as team. So this talk is really about a project I kind of started as a postdoc and then put down and then picked up again. And this is kind of how science goes. You get distracted by some things and then you come back to other things. And so I was really interested in looking at calponent homology domain. So at the time I was studying another protein that had a calponent homology domain. These domains are classic cytoskeletal binding domains. And I just did a quick bioinformatic search looking for proteins that might have CH domains in Drosophila genome. And there, an uncharacterized protein popped up. It was CG1366 or something like that. CGs are just uncharacterized Drosophila proteins. And I was like, huh, that's interesting. Like, this is a protein that has a CH domain. It's likely a cytoskeletal interacting protein, and it's uncharacterized. So let me clone it and see what it looks like. So I cloned it, transfected it into cells, and it's also known as split discs, and it happens to have a very interesting human homologue and a story associated with it. So again, by chance, just looking at CH domains, found this protein that's not well characterized, and then it happened to have a really interesting story with humans. So the human homologue is known as SPECC1L, which is a terrible name. I think it's sperm antigen with coiled coil-like domains 1. And here's just a diagram of the protein. And it's several coiled coil domains, and then at its very C terminus, it has this calponent homology domain. And as such, and as would be predicted, it is putatively a cytoskeletal interacting protein. But interestingly enough, it's been associated with a spectrum of cranial facial disorders, including facial clefts, wide eyes, and incomplete closure of the esophagus. And these two particular mutations, this Q415P and this G1083S, were unfortunately associated with a family all having some of these cranial facial disorders and pathologies. And why they seem kind of desperate in their pathologies, they actually all are kind of related to cranial neural crest cell migration. So all these phenotypes really highly suggest that there's something happening during cranial neural crest cell migration that's leading to these pathologies. And if you're not familiar with cranial neural crest cell migration, I certainly was not, because remember, I started this whole idea of looking at calponent homology domains, and now I'm talking about cranial neural crest cell migration, but that's exactly how science kind of works. Cranial neural crest cells delaminate from the neural epithelium and then migrate as clusters to form a large, they migrate using a distinct path, and they do this during, they undergo an EMT-like transformation, but they are in constant contact with one another during this type of migration. So this is an example of collective cell migration. And they emerge from the hindbrain and migrate through a variety of extracellular matrix environments. And that's really also important because their adhesion to the extracellular matrix as they're migrating from this neural tube closing to their craniofacial origins is really important. They experience different types of environments during this process. And then as such, they are really critical to forming a lot of different types of facial structures, including bone, and again, cleft palate is just a issue with the two parts of the face actually fully closing. Same with esophagus and wide eyes that happens. So they're really responsible for a number of facial and cranial structures. And again, if their migration is aberrant, you do have abnormalities. So in mammalian cells, this protein SPECC1L co-localizes both to actin and microtubules. So in the top panel, we have a GFP tagged form of this protein, and we're looking at tubulin. And the bottom, there is an antibody to this human protein as well. And now we're looking at actin. And so this was all happening while I was just looking at this Drosophila protein, and then had to dive into this literature to figure out what's going on with the human protein. But what really made me go, oh yeah, so then they collect, so my literature dive did bring me to a Drosophila paper that sort of started to characterize this protein as well. So the Drosophila homolog of SPE1L is this protein, again, that I found with that bioinformatic study. And the amorphic alleles are not completely lethal. Some of the escapers have a variety of phenotypes that are most notably cleft proboscis and wings. And that's kind of interesting because, again, flies don't have neural crest cells. Neural crest cells is a mammalian thing, but it's just kind of interesting that they have cleft proboscis and how it sort of parallels what happens in humans, unfortunately. But what these phenotypes do suggest are aberrant migration and cell migration, aberrant adhesion and cell migration as well. So it seems that even in flies, despite the fact they don't have neural crest cells, this type of this mutation in this split discs, which is the human homolog, which is the fly homolog of SPE1L, is causing adhesion type phenotypes as well. So what made me go, that's funny, is that when I cloned split discs and I transferred it into S2 cells, it seemed to closely associate with non-muscle myosin 2 rather than microtubules. So on the left, I have, again, the cell-expressing EGFP spaghetti squash, and on the right, M-cherry split discs. And to me, this looked like really, really good localization between non-muscle myosin 2 and split disc, which is what was not reported for the mammalian homolog. I went even further and looked at microtubules at the same time, and again, I'm not really seeing any sort of interaction between microtubules and split discs. It's more split discs and non-muscle myosin 2 and actin. And again, when I was a postdoc, one of the projects that led me down this sort of rabbit hole of understanding this protein was the fact that I was studying a microtubule actin cross-linking protein. So the microtubule actin cross-linking protein I was studying also had a CH domain, which is why I was interested in the first place. So to me, and my experience of understanding actin and microtubule cross-linking proteins, this did not look like one of those to me, even though that was reported from mammalian homolog. So this made me really perk up because this is interesting. Why is a localization different? And what is it doing localizing with non-muscle myosin 2? If we look at the dynamics of the protein, it pretty much mirrors that of non-muscle myosin 2. And again, this is a movie of an S2 cell by Turf. And we're looking at split discs in red, spaghetti squash in cyan, and then the merge of the two. And so even the dynamics looks very similar to me as non-muscle myosin 2. And I also looked at the localization of split discs with tropomyosin. Though this is a lower expressing cell, tropomyosin is another protein that sort of co-segregates with non-muscle myosin 2. It protects and allows actin filaments to bind the non-muscle myosin 2. And again, we're seeing a very similar localization pattern between split disc and triple myosin, again, really highly suggesting that this protein is interacting in some way, in some form with non-muscle myosin 2. And moreover, if you get rid of the heavy chain of non-muscle myosin 2, which should disrupt non-muscle myosin altogether, we also see a disruption of split disc. So it seems like it's traveling with non-muscle myosin 2. And we can also disrupt it using RNAi against the essential light chain. One thing that's really beautiful about Drosophila cells is that they are really sensitive to RNAi. So you can basically knock down almost any protein that you want just because of the nature of RNAi in these cells. So again, it looks like this protein is doing whatever non-muscle myosin 2 is doing. If we disrupt non-muscle myosin 2, its localization is also disrupted. In fact, I have gone through several other myosin-associated proteins and tried to display them or disrupt them, and it seems in every case, whenever non-muscle myosin 2 is disrupted, we see disruption of split discs. I've yet to find a condition where I can uncouple the interaction between the two proteins at this point. So they're really closely associated. But the question is then, if they are co-localizing, how is it co-localizing with non-muscle myosin 2? And so to do this, my students and I got together and decided to use this Mander's coefficient of overlap. This is a way to basically determine if two proteins are sort of localizing in the same place at the same time. Mander's overlap coefficient expresses the degree of which two proteins are structurally spatially overlapping and takes into account the total abundance of the fluorophore that overlaps. And it results in values from 0 to 1. So I'm telling you this because I'm going to show you a series of graphs and I want you to be able to sort of follow along. And high values for both of these graphs, of these values suggests some sort of degree of co-localization. So on the left, you would have a discrete protein and yet another discrete protein, and they both are sort of in the same place and overlapping, and so we're seeing a high Mander's coefficient for both of them. Where on the right, we have a discrete protein in cyan and maybe like a soluble, maybe GFP in another. And so you can see how you'd have a high value of co-localization for one, but a very low value for the other. The low value for the GFP that's sort of spread out across the cell, whereas a high value when they're, the GFP would probably discretely co-localize with our other discrete protein, giving us a very uneven Mander's coefficient. So we're going to use this, coefficient to try to track split disc and non-muscle myosin 2 co-localization. So this is the wild type version with, the top panel is wild type split discs, and then the bottom panel, we have deleted that CH domain, which got me started on this rabbit hole. And what we found is that if you delete that CH domain, we do begin to uncouple that co-localization between split discs and non-muscle myosin 2. We also deleted the coiled codomains, the first coiled codomain and the first and second coiled codomain, and deletion of those two coiled codomains really did nothing to prevent the co-localization between non-muscle myosin 2 and split discs, but it was really deletion of that CH domain that started to uncouple the localization between these two proteins. And then we also have the opportunity to look at the analogous mutations to those that causes the cranial spatial defects in humans. And so those mutations are Q266P and G915S. So this protein is so conserved in flies and humans that we can find the same exact residues that causes disease in humans. And so then we wanted to look at their localization with non-muscle myosin 2. And interestingly enough, and probably not surprising given what I just showed you about the CH domain, if we express split discs and either wild type or these two mutants with non-muscle myosin 2, we see that the mutation in that CH domain, that G915S, does uncouple the localization between split discs and non-muscle myosin 2. So again, that whole CH domain, which started me on this rabbit hole, seems to be really important for the localization of this protein with non-muscle myosin 2. But what about cells that migrate? here is a movie I pulled from this paper of cranial neural crest cells migration, and you can see that they are starting their stereotypical path and that they're constantly in contact. And again, as I said before, one thing that's really important is that they have to go through a variety of substrates on their way to making these structures. And the pathology that is associated with this protein, SB1L split disc, is caused by these mutations rather than deletions. So we really want to know then what is the role of non-muscle myosin 2 in migration, and how does split disc maybe possibly intersect with that? So non-muscle myosin 2 plays a really key role in the formation of focal adhesions. Focal adhesions are these cellular complexes that, that actually connect the actin cytoskeleton to the extracellular matrix. They're thought to be layered in several different sort of protein family groups. We have the integrins, which are actually interacting with the extracellular matrix. These are receptor proteins that then bind to a signaling level. That signal level then binds to a force transduction level, and then that couples with the actin cytoskeleton. This gives the cells the ability to grip during migration. And here we have Drosophila cells. These are D25 cells. This is an epithelial-derived cell line that it's expressing the Drosophila vinculin. And the bright puncta are these focal adhesions that are forming. And again, this is, again, imaged by turf microscopy, which happens to be my favorite way of imaging a lot of things. So the defects that we've seen in craniofacial structures might suggest that it's a aberrant cell migration that is the problem. And again, if you have to get to point A to point B at a specific time during development, and your trip is delayed by any amount of time, that might lead to the inability to form the proper facial structures needed. And so our hypothesis was that if split disc plays a role in non-muscle myosin 2 regulation, it's possible that it's actually focal adhesions that are going to be affected. So what we did was a whole bunch of focal adhesion tracking. This is work really done by JJ, and I basically said, figure this out, and he did. And so we took a lot of movies, and then we started deleting these proteins. We deleted depleted split discs, spaghetti squash, and NBS, and then tracked focal adhesion turnover. And interestingly enough, what we found was that there was no difference in the focal adhesion assembly dynamics when we deplete these different proteins. But we did see a slight increase in the disassembly rate when we depleted split discs. So no difference in the assembly of focal adhesions, but we did see an increase in the rate of focal adhesion disassembly following split discs RNAi. But again, the disease isn't caused by a depletion of this protein. It's actually caused by these point mutations. So we really wanted to drill down, what are these point mutations doing? So we're able to knock down split discs and then add back a version with these point mutations. And then we were tracking the focal adhesion dynamics on top of that. And so the N is small because these experiments are kind of tricky. But what we found is that there's no difference in the focal adhesion assembly rate, which is actually not surprising. We didn't see any difference when we depleted the protein. But what we did see was a huge increase in the rate of focal adhesion disassembly rate when we expressed that Q226P mutation. So what's really interesting about this is that that Q266P mutation did not disrupt the co-localization between split disc and non-muscle myosin 2 at all. It was the G915, which looks like it has no effect in terms of focal adhesion disassembly dynamics. But expression of this Q226P mutation caused a 1.75 increase in the disassembly rate by our tracking. So what about actual cell migration? So this is probably a 6-hour movie of our D25 cells taken by phase contrast microscopy. And we're able to track, again, JJ did this Herculean task of tracking all these cells. And we knocked down, GFP is our control here, and we knocked down split discs RNAi, and we tracked tons of cells. And we found a slight but statistically significant decrease in the rate of cell migration. Here's where this is really interesting to me, because we're seeing an increase in the rate of focal adhesion disassembly, but decrease in their ability to migrate. And so trying to connect these two has been a challenge, at least in the lab, and we have a lot of discussions about this because, yes, focal adhesions are really critical cell migration. One would think that if you're increasing your disassembly rate, you would actually increase cell migration. But in fact, maybe what is happening is these cells aren't able to make the proper grip when they're migrating, which might be slowing them down. In terms of what's happening in the human pathologies, indeed, if SPC1L is contributing to focal adhesion dynamics in cranial crest cell migration, and it's increasing the rate of focal adhesion disassembly, that might be enough to mess up the timing of these cells during their migration, leading to these pathologies. Again, this is wild speculation, but this is what these results are suggesting right now. So in the future, we're setting up these experiments, again, really tricky. We're able to knock down the endogenous version of split discs and then add back these point mutations to see if how they will affect cell migration. But that is TBD, again, as those experiments are kind of tricky. So we really are trying to focus on this idea of how do these potential adhesion phenotypes translate to defects in cell migration. And here's where I started to collaborate with many physics students, which is wonderful about working at a small liberal arts school, and it's just across the hallway, the physics department. So I've able to convince a couple physics students to come to the squishy side and use their quantitative skills in my lab. And so the technique that we've been using is an old technique. It's been around for a while. It's called traction force microscopy. And what you're looking at, and you can't see, but what you're looking at is a cell that you can't see on a compliant matrix. And that matrix has embedded in it fluorescent beads. And so as the cell pulls on the squishy matrix, it is displacing the beads to some degree. And what we can do is back calculate the forces that are caused by this displacement of the beads. The compliant matrix is made of polyacrylamide, and we know basically the Young's modulus for this, the spring constant. And so we can use that to actually back calculate forces. I am by no means a biophysicist, but again, that's why I've convinced several physicists to participate in this project. And so this summer, my student Sushruta was able to do a couple of these experiments, and this is kind of what the data looks like. So we see some vectors showing exactly where the forces are being displaced, and the beads are being displaced, and forces are being manifest. And then we have a heat map here. And so again, a very small n, because we're just getting this up and running, and these experiments are a little bit tricky. But what it looks like is that when we get rid of split discs or knock down, again, the 3' UTR split discs, we are seeing an increase in the force. So again, this is really interesting to me because the canonical thinking is like if you increase forces, actually what you'd be doing is causing those focal adhesions to get bigger, stronger, more robust, and then as such, harder to actually disassemble. But what we're seeing is that our focal adhesion disassembly rate is increased, but maybe, possibly, and this is again really early on, we're seeing increase in forces. So still a lot of contradictions we're trying to sort of fit together, try to understand what's really happening in this story. And what we are also gearing up to do, because it's really apparent to us, is that we have to look at these disease-causing alleles, because those seem to be the more important aspect of this story. So we're going to be gearing up to do these experiments now where we can, again, deplete the endogenous split disc and then add back the disease-causing point mutations and test that process. I'm also really interested in understanding what else split disc is interacting with and why. And so in order to do this, we set up a proximity ligation and mass spec experiment. And this is with collaboration with Peter Bar-Gillespie at OHSU. So essentially we have made a split disc variant with a beer biotin ligase instead of a GFP. And so theoretically what this would do is any protein that came within sort of some closer proximity to split discs would get biotinylated. then we can use, we can lyse those cells, put them on a streptavidin column, and then pull out theoretically any of the proteins that would interact with split discs. And then do a mass spec. And so we've done 2 runs of this, and this is kind of the breakdown. And we're still trying to wrap our minds around what we have here. My student sort of broke these up into different sort of functional categories, I guess Go terms really, but different functional categories. Not surprising, a lot of ribosomal subunits. I don't want to ignore those because there could be some interesting biology there, but that's also kind of to be expected given that ribosomes need to make the protein. But then we found some interesting cytoskeletal proteins, some proteins involved in endocytosis, DNA repair, mitosis, development, RAS signaling, and others. Interestingly enough, there is not non-muscle myosin 2 here. And so I did this experiment because I really thought, okay, yeah, this is a way to actually find exactly which polypeptide, non-muscle myosin 2 is a hexamer, which one of these proteins is split disc really associating with. And to my surprise, it did not come out in our runs. What is on this run was MBS, which is a phosphatase for non-muscle myosin 2. So there's that. And a couple other, again, cytoskeletal and even focal adhesion proteins. RIA is talin in humans. So there is some interesting interaction between split disc and the focal adhesion, possibly, and actin, but not non-muscle myosin 2, which is, again, really perplexing. So there's a lot of contradictions and interesting results that we are still trying to fit together. But I'll conclude that split disc co-localizes with non-muscle myosin 2, and we know that it's that CH domain that's needed for this co-localization. And again, that point mutation to that G915 to serine also disrupts the interaction with non-muscle myosin 2. If we deplete split discs, we do get a slight increase in the rate of focal adhesion disassembly, but moreover, it's expression of that Q266P variant that further increases the focal adhesion disassembly. Despite this increase in focal adhesion disassembly, we do see that it decreases the rates of random cell migration. And again, this is somewhat of a contradiction. In my simplistic view of this, if you increase your deadhesion, you probably would increase your migration, but we're seeing the opposite. And that this depletion may also lead to increases in traction stress. So again, very strange sort of set of results here. One, again, would assume, and the most simplistic sort of hypothesis here is that if you're increasing traction forces, you're going to be increasing at least the size and the robustness of focal adhesions, as they do depend on non-muscle myosin 2 to sort of pull on them to increase their size. So again, want to thank the people that do this work. I have a band of undergrads that are really fierce and thirsty for knowledge and really work hard. And this is back when we could actually be in the same place at the same time. So I long for these days. And I want to thank past lab members, my collaborators at OHSU, and Dr. Greta Glover, who was our instrumentalist, who's really been helpful in helping me train physicists who usually don't have pipette or lab skills, biology lab skills, to be brave and leap into the squishy world. Also my source of funding, National Institute of Health and Reed College. And this is, again, a beautiful picture of this canyon that runs through the campus I caught on my way into work one day. And I will happily take any questions if you have any. Oh.
All right, well, first question looks like it's from Ethan.
Hey, amazing talk, Eric. It's awesome. Thank you so much. I have a couple questions. So in the beginning, when you showed the mutant phenotype of the Drosophila, they seem to have tissue-specific defects, right? So like, I don't know anything about Drosophila cell, right? But like, where do they come from? Do you know, are they from like the tissue that are like defective? Or? Yeah,
so the wing blisters are really, really similar to what you see in adhesions and integrins. So they almost phenocopy mutations in integrin components in the wing disc. So that's another reason why I've been focusing on integrins and focal adhesions, because that pathway is very similar with, when the mutant split discs in the wings is very similar to what you see when you get rid of integrins. The proboscis is again another adhesion phenotype. That might be more of a cell-cell adhesion phenotype, which also kind of makes sense. But I haven't really drilled into other mutations in that pathway that causes split proboscis and flies. But definitely integrins is a dead ringer. So that same sort of phenotype happens with mutant and integrins.
Right. And split disc is not expressed. There's no tissue-specific expression. It's expressed everywhere, right? In the animal. Yeah.
Yes, but at different levels. So I can tell you about cells. I can't tell you exactly about tissues at this moment. But for instance, the D25 cells that I was doing some of the migration assays with, they actually have a mild to low expression level split discs. But then I have another cell line that is derived from the central nervous system that expresses it a lot. So there is a low level of expression kind of everywhere, but then there are some tissues that have very high expression. If you think back to the flies, I'd have to go back and look and see what tissues have high and low expression of this protein. But I can tell you that it is variable, at least among cells. Yeah.
Thank you.
I should have made the proclaimer. I am a Drosophila cell biologist who dabbles with flies. But yeah.
All right, we have another question from Brooke. Go ahead, Brooke.
Okay, I have a couple questions actually. Did you try the BioID with the mutant, the Q266P mutant?
No, that is a great idea. I did not. That was just with wild type full length.
Okay.
Yeah.
Yeah, it seems like that is probably disrupting some interaction that's not with NM2.
Yeah.
That would be super interesting.
That is, yeah, that's a good idea.
And then I also had a question of, do you think your focal adhesion disassembly could be kind of incompletely disassembling them? Like if you're looking at vinculin, do you think that the integrins could be left behind and that's why you're kind of seeing a decoupling of the cytoskeleton, but the integrins maybe still are connecting with the substrate?
Yeah, that's also a good point. You know, part of the problem with doing some of these experiments is you only look at a couple, proteins at a time, and there could be some decoupling, you're right, of some of the focal adhesion complexes. I have no clue about that. But that's another good point. It could be that there's just disrupting it in different ways. Yeah.
Great talk. Thank you.
Thank you.
Oh, sorry, I was muted. The next question is from Joel. It's my bad.
Okay. Really enjoyed your talk, Derek. So I'm wondering about this microtubule association seen in the mammalian version of this thing. Is it possible this thing is somehow connected to both kinds of cytoskeleton? Under some circumstances, the microtubule association might be seen, but not conditions you've looked at. Has it been looked at comprehensively enough, for example, in the intact animal or embryo or something to address that at all in flies? And related, do you know the localization in the absence of the CH domain? Were there any microtubule associations that might have been detected in your proteomics study?
Yeah, so I'm looking at that right now. TAC is microtubule associated protein. This protein called bifocal and microtubule associated protein, and obviously dynein is a microtubule associated protein. And so
those came out, sorry, to be clear, that was from the proteomics?
Correct.
Okay.
Yeah, and I agree. I think my initial into this was oh, I think they're wrong, and I'm gonna show them how they're wrong. But in fact, I think you're right. It could be that these conditions might be either happening in the animal, because I haven't looked at this in the animal. And the group that did the first characterization of this protein were the ones that named it split discs in flies. They did sort of they looked at it in flies, they looked at it in zebrafish, and they were also relating it back to this human disease. And so the amount that's known about flies, I already showed you. So no one has really looked at it in the tissues, as you suggest. I mean, that's another reason why I'm trying to come at it with different angles using different cell types, because I've showed you S2 cells, which are the workhorse of most fly cell biology, but I'm trying to use these other different epithelial cell lines. I'm using a neuronal derived cell line because I do want to make sure that I want to catch it from all angles and make sure that I'm trying to get a comprehensive view of what this protein is doing. Excuse me. That being said, I still have not seen any specific microtubule association with it. But the other sort of aspect of this is there's a lot of endocytosis. So one of the other, the assembly of focal adhesions also involves endocytosis. So it really could be that it's helping drive microtubules to the focal adhesions to cause them to be endocytosed. So I'm sort of coming back around to maybe thinking, all right, maybe there is some microtubule association or interaction that I've been sort of being blinded to by my own biases. So I think it's a good point. I haven't seen anything yet, but I'm going to circle back around to it because I think that's what the biology is kind of saying.
Yeah, and I guess it's conceivable. That's sort of what it's suggesting, that maybe in some of the mutants you'll actually see that association, because if it's switching, for example, it may be if you delete the domains that are involved in interaction with the actin-myosin system, it uncovers the microtubule association.
That's a really that's a really good point. Yeah, and so I don't know. I have not done the experiment where I've looked at the CH domain specifically with microtubules. Certainly I'll tell my student to get on that now. But yeah, thank you.
Great, thanks.
Okay then. Thank you very much, Derek.
Thank you.
All right, so now we
maybe says to remind everybody that Derek will be back again this afternoon.
Oh yes, sorry. Derek will be back this afternoon doing the Q&A with C. Brandon Obunu. So be sure to catch that. Yeah, thank you very much, and we look forward to seeing you again.